No. Hi everyone, good afternoon guys. Welcome back to Unacademy NEET English channel, and I am your biology educator. So yes guys, finally that D-Day, that final day is here, which we were really waiting for. Should not have come, but it is here as another opportunity for us to prove again that yes, we can do it. Now, two things. In today's session, we are going to revise very important topics, those topics which are 100% going to appear in your exam. Like those questions, those topics which are never skipped by NTA. So make sure that you revise these things with me. This is your final revision, which can ensure 360 out of 360. And also have that faith that you already remember a lot of things. This is just a revision of all those topics that we have already discussed in our marathons, in our NCERT, but just a final touch before we go for our exam. I hope everything is ready. You're ready with your proper dress. You've got your admit card downloaded and all those things are ready. If there is any doubt, please let me know in the comment section. I will answer to that. And yes, the most important part of a preparation, guys, to be mentally ready for the exam. Make sure that before you go for the exam, you still take these tests. It is Friday, Saturday, right? Sorry, this is Thursday, Friday and Saturday. We still have 3 days. 3 days in which we can take at least 6 tests or at least 3 tests. So at least take these 6 overall tests or 3 major tests. Take these tests, and on the basis of these tests, now revise those things. I am going to revise these things with you on the basis of what has always appeared in the exam without any skip in any year. So these are those topics, but you have to find your weakness on the basis of these tests. Make sure that you do take these overall tests before you go for the exam. Get mentally ready for the exam, right? Now on that note, give me a thumbs up that you're all set for the final revision and you're all bugged up. You all are totally excited about the test. Yes, there is anxiety, there is always fear of the exam, but yes, you have to feel confident before the paper comes in front of you. And even if something goes wrong, see, we cannot control all the situations. If something goes wrong, you will not lose heart, you will not lose your confidence, because whatever is happening with everyone. Be sure this time paper is not getting leaked, because that will be the height of anarchy, but that's not there yet. We are not there yet. So make sure that you are ready. Give me a thumbs up that you will be taking these at least 6 overall tests before your exam. Give me a thumbs up that how many of you are ready, and we'll start with the final revision. Yes, guys. Yes, everybody. Let me know, guys. Quickly. Quick. Yes. Title has 36 instead of 360. I'll get that. That's really a mock. No. 360 out of 3. Oh. The title has 36. Don't worry. Everybody is working day and night, guys. The team is actually working day and night more than us. The team working behind the screen is always there, up, going through the WhatsApps and the notifications. They are the ones who notify us. So just ignore that. You will be getting 360 out of 360 if you prepare till the end. Yes? All right. Take it as fun and let's revise, guys. Let's start with our revision. Let me see your chat. Are we all ready? Genuinely, how many? You will see all these questions. Let's start from here. These are the topics which are never skipped by NTA, guys. I have taken some clips from NCERT, some clips from our marathon sessions which we have done, the flow charts that we used, right? So let's start with decomposition. I'm starting from ecology because this time also ecology got maximum credit. right? Maximum questions from ecology. So we are only going to discuss few topics from every chapter which are never skipped by NTA. That's what I'm telling you. All right. Okay. So starting with decomposition, guys. Decomposition. What are the 5 steps you have to remember? Fragmentation, catabolism, leaching, humidification, and mineralization. Now, the raw material for decomposition is known as detritus. It includes plant waste as well as animal waste. Now, there are 3 steps which will occur simultaneously. One is fragmentation, catabolism, and leaching. Fragmentation is breakdown of large particles, detritus, into smaller particles. It is done by the action of these detrivores like earthworm. Second is catabolism, in which detritus, which has been broken down into small particles, is further broken down into simple inorganic molecules. This process is called catabolism, and who does that? Fungal and bacterial enzymes. Is that clear? Right. And then leaching. Leaching is the percolation of water soluble inorganic molecules deeper into the soils, and it becomes unavailable, becomes precipitated there. Now these processes will occur simultaneously. That is why I've written it here. And then humidification. After catabolism, guys, humidification will take place. Formation of humus. Now what are the properties of humus? Dark colored, amorphous, colloidal, resistant to microbial action, undergoes slow decomposition. And now from this humus, very slow release of nutrients into the soil is known as mineralization. These are the 5 steps. What are the very important factors affecting the decomposition, guys? Remember, decomposition is aerobic process. Oxygen is necessary. If it will be anaerobic condition, decomposition will not occur or will be very, very slow. Then if chitin and lignin are present, again decomposition slow. But if sugars and nitrogen rich compounds are present, then your decomposition will be faster. And the most important factors are temperature and soil moisture. Higher temperature and higher humidity will support decomposition. Please let me know that you're ready with this topic. Next point is, guys, population interaction. Population interaction, see here. Mutualism. We know that there are different types of interactions. Mutualism is positive, benefits both. Commensalism benefits one and unaffected other organism. Then in parasitism, one is benefited, another is harmed. Predation, one is benefited, again another harmed. Competition, both are harmed. And in amensalism, one is harmed and other is unaffected. So all those which are, so this is overall positive interaction. These are overall negative interactions. You have to learn the examples, which example is suited or which example shows which type of interaction. So I have tabulated these examples for you. I have brought many tables for you. Earlier my plan was to share all these on the Telegram channel so that you can quietly revise them, but now as we know the situation, so please make sure that you keep taking the screenshots, guys. Keep taking the screenshots. See, in mutualism, lichens, rhizobium in the legumes, right? That is symbiotic association. Legume root nodules, corals, and then mycorrhiza. These are all mutualism. Then plants and the pollinators, that is also mutualism. Then epiphytes on the trees, barnacles on the whale, cattle, ergot and the cattle. So this is all commensalism. So make sure that you learn all these examples. Take it. It's not going to be a marathon session. I am quickly going to revise these topics. You also keep writing down the list of the topics. Make a list of the topics, guys, which you have to revise. And other than that, I'm telling you, you will have your personal shortcomings. How will you identify? Make sure that you take a test. Make sure you take some tests. Now, population growth models. See, there are 2 types of growth models that we have studied. One is the exponential growth model and second is the logistic growth model. So, exponential growth model gives you J-shaped graph, J-shaped curve, and this is the logistic growth model which gives you, will give you, yes, this S-shaped graph or sigmoid shaped graph. Every year we have got questions from this topic. Every year. You don't believe me? Check your PYQ book. Every year we are getting questions from this topic, guys. So now see, Here the equation for exponential is dn by dt is equal to Rn, and for logistic it is dn by dt is equal to Rn k minus n upon k. Also know what is this k? K is the, what is k? Let me know in the chat. Yes, everybody. What does k denote? What does k denote, guys? You have to let me know in the chat or in the comment section. If you don't know the answer, I'll be asking you lots of questions. If you don't know an answer to the question, please make sure that you pause the video, you look for the answer, and make sure that you incubate over it, because that question might come in the exam. Now, coming to the next, production and productivity. They've asked the definition, they've asked the units, even in the previous year. See, what is production? It is the amount of organic matter produced per unit area in a period of time by plants during photosynthesis. Now, units are very, very important. It is organic matter. See, it can be in terms of weight or it can be in terms of energy. In terms of weight, it is gram per meter square, energy kilocalorie per meter square. Now, what is productivity, guys? What is productivity? When we talk about productivity, it brings the time factor. So here, just note down the units here. The unit of production is gram per meter square, while unit of productivity is gram per meter square per year. Similarly, kilocalories per meter square per year. So this productivity will help you compare the efficiency of productivity of two different ecosystems. We have two: gross primary productivity and net primary productivity. Gross is what is produced by the organism, by the plants, and net is what is available to the herbivores, to the next one. Why? Because whatever is produced is not entirely for the next trophic level, it is also utilized by the organism for its own respiration. So we minus R, that is respiratory loss, and we get NPP. Are these points clear? Be very, very sure about the units. Is it clear? When it is without time factor, it is production. With time factor, it becomes the productivity. Now, there is not even a single year where we have not got questions from this topic, the evil quartet. Yes or no? So we know what is the evil quartet. These are the 4 reasons, 4 causes of biodiversity loss. And you must remember, out of all these 4, 1, 2, 3 and 4, the most important is the habitat loss and fragmentation. This has been a direct question, guys. Now see, habitat loss and fragmentation, most important reason, most dramatic example is loss of tropical rainforest. And tropical rainforest, which was earlier 14%, has now reduced to, it has shrinked to only 6%. They are known as lungs of the planet. What is the cause of this habitat loss? Like in tropical rainforest, Amazon forest, soybean cultivation for raising the beef cattle. These are the 2 reasons. Please tell me that this point is clear. Second reason is over exploitation. Need becomes greed. Species have become extinct in last 500 years. Very a lot of, like, that extinction rate has increased a lot. Names, look at these names, and these names have come in match the following: Stellar sea cow from Russia, passenger's pigeon, dodo from Mauritius, quagga from Africa, thylacine from Australia, and 3 subspecies of tigers named Java, Caspian and Bali, depending upon the place they belonged to, right? Then you should be knowing their countries that they come from or they were present in. Stellar sea cow in Russia, dodo Mauritius, quagga Africa and thylacine in Australia. Is that clear? Now third is alien species invasion. 3 names which are very, very important: Parthenium, lantana and Ishornia. So because of these alien species, the native species are becoming extinct, right? For example, Nile perch, an alien species from Lake Victoria, it led to the extinction of 200 species of cichlid fish, Clarius garipinus, to the extinction of indigenous catfish. Then co-extinction. Co-extinction is always shown by those organisms who are co-dependent on each other. So they co-evolve and get co-extinct also. So mutually co-dependent species showing in obligatory manner, which are dependent on each other in obligatory manner. So pollinators and the plant species, parasites and the host. So out of all these 4 reasons, guys, habitat loss is the most important reason. Please tell me these points are clear. Another very important, very important, yes, screen is not clear. Just a second, just a second. Just a second. Let me check this one. Yeah. Give me a second, guys. Give me 1 second. Just a second. Becoming better, right? Give me 1 second. Yes, guys, wait a second. Is this better? Yes? Now it is better, right? All right. Let's do with this. Is this clear? Now? Right. All right. Moving on to the methods of biodiversity conservation, guys. Now, methods of biodiversity conservation, little more, ma'am, don't use green color. See, these are already made. Please adjust and now understand. I've already written it. Methods of biodiversity conservation. What are the 2 methods? One is in situ and second is ex situ. Right? Now, in situ conservation involves biosphere reserves, national parks, sanctuaries, sacred groves and hotspots. While here in ex situ we have zoological parks, botanical gardens, wildlife, pollen banks and seed banks. What are ex situ? When the organism is taken out of the habitat for the conservation. Is that clear? This is off-site conservation while this one is in-site conservation. Are these points clear? You will get question from this topic. You will get this question from the topic. Look at the number, the years. I have tried to write some years also. Now, let's come on to biotechnology. So, BT crops. So, we know that BT crops have a gene. They carry a cry gene from a bacteria named as Bacillus thuringiensis. Yes, from Bacillus thuringiensis. We know that this cry gene is responsible for the production of cry protein. The name of the protein starts with the capital letter and the cry stands for crystalline. We also know that this crystalline protein is non-toxic when produced. That is why it will not harm the bacteria Bacillus thuringiensis. Therefore, not harmful to the bacteria. Right? Not harmful to bacteria. But guys, as soon as it gets the alkaline medium, what happens? It becomes toxic, it becomes active. So this is protoxin in active form, while this one is the active form. So where does it get the inactive? Where does it get the alkaline medium? This alkaline medium is found in the mid-gut of insects. So we have understood this mechanism, right? So therefore we took out this gene from the from the bacteria, put it in the crops. When the crops are eaten, these crops, bt crops, are eaten by the insects, the midgut of insects which has the alkaline medium, this is where it becomes this cry protein, it becomes active and causes lysis, becomes active and causes lysis of midgut, epithelial cells of midgut, epithelial cells of midgut. Is that clear? Please tell me if you are clear with it. Now, what are the three orders of insects? Orders of insects against which this cry gene is effective. Orders of insects. Yes, guys. Let me know the orders of the insects. I'll tell you that cry gene comes in various forms. There are three forms that are given. One is cry 1 AB, cry 2 AB, cry 2 AC. Yes, guys. Everybody, please quickly let me know. Everyone. Yes or no? Quickly let me know, guys. Is that clear to all? So cry 1 AB controls the corn borers. Cry 2 AB and cry 1 AC. This controls the cotton bollworms. Yes, guys. Who will tell me? Okay, I will not use a green pen. I'll not use the green pen. Yes, fine. So orders of insects. Which are the orders of insects, guys, which are effective or which are killed by this cry gene? Yes. So it is Lepidopterans, it is Orthopterans, and it is Dipterans. Yes or no? So you have to remember doll. So learn it this way. So Dipterans, Orthopterans, and Lepidopterans, and you have to know the names of those organisms. Yes or no? So just learn the doll. Lepidopterans, Dipterans, and yes. Is that clear to all? Is that clear to everyone? Wait, let me just remove this. I think now it will be clear. Is this green color clear now? Right? Yeah. Now it is fine. Clear to you all? Is this clear? Better? Chroma key done. Yes. That was the problem. Because last time I could not take class on this screen, so I had to use that chroma. So that setting wasn't changed. Is that clear to all? Now guys, let's talk about human insulin. See, every year we are getting question from this topic, human insulin. So we know that when human insulin is produced, it is produced in the form of proinsulin. So beta cells of pancreas, of isles of Langerhans, they produce what? They produce proinsulin, and proinsulin contains three polypeptide chains, right, which are A, B, and C. While later on it is modified to form insulin. So proinsulin contains three polypeptide chains, and insulin, it is modified to form insulin, which contains two polypeptide chains. which is just A and B. So basically during the modification, what are we doing? During modification, we are removing the C peptide chain. We are removing the C peptide chain. So this is A, this is B, and this biggest chain is C, which is removed during the modification. Is that clear? So which was the first company to successfully produce human insulin? So before human insulin was produced by biotechnology, it was pigulin, which caused lots of allergies. It caused lots of allergies. But later biotech gave us human insulin, and that was known as humulin. So humulin means the human insulin produced by biotechnology. Is that clear? So which was the first company, guys? It was Eli Lilly. Let me know the year in which this American company was able to successfully produce humulin. Yes, guys, let me know the year. What did Eli Lilly do? Yes, please. What did Eli Lilly do? They took two different E. coli and two separate, and in one they kept the put the A gene, and in the other they inserted the B gene, and then they got both the polypeptide genes were produced separately. So what was the overall philosophy? Produced the two polypeptide chains separately, right? Removed them, extracted them separately, produced separately, extracted separately, and then combined artificially, combined by artificial disulfide bonds. Yes or no? Artificially by disulfide bonds. Yes, guys, is that clear? Combined artificially by disulfide bonds. Please tell me that this point is clear to all. Is that clear? Right? All right. So this was human insulin. Now talking about PBR 322, every year we are getting the question from this topic, guys. 26, 25, 24, 23, 22. We are getting this question every year. Now see here. So what is PBR 322, by the way? PBR 322 is a cloning vector, an artificially produced cloning vector. Yes. So what are the properties which a cloning vector should have? What are the properties a cloning vector should have? Yes, please. The three properties that they must have. One is ori, from where the replication begins. So any foreign gene attached to ori will obviously replicate and also controls the copy number. Second is the selectable markers. What are the selectable markers which help you identify between transformants and the non-transformants? Which are the selectable markers? Generally, the selectable markers are antibiotic resistance genes. Antibiotic resistance genes. Yes. Sometimes it can be a chromogenic gene for chromogenic substance also. Gene for chromogenic substance. Please tell me these points are clear. And the third, it should have single cloning site, because more than one cloning site will complicate the process. Yes or no? These are the three features of any cloning vector that we use. Now we know that in plants we use Agrobacterium tumefaciens. Yes or no? For plants it is Agrobacterium tumefaciens. Agrobacterium tumefaciens. Yes or no? I'm revising everything here because it has this TI plasmid which contains tDNA. This contains tDNA. Yes or no? And in animals, guys, the favorable vector is a retrovirus. Now, when they have to be used as a cloning vector, both of these are made non-pathogenic, because otherwise they are both pathogenic. We have to first transform them into non-pathogenic. Is that clear? Now, similarly, we have an artificially produced cloning vector, which is PBR 322, plasmid Bolivar Rodriguez, and 322 is the number. Now, look at the structure of PBR 322. Which are the clone? See, this is ori, first point. Second, it should have cloning vectors. Sorry, it should have selectable markers. These are the selectable markers. Ampicillin resistance gene, tetracycline resistance gene, both are antibiotic resistance genes. Yes or no? Which are the molecular scissors used for inserting a foreign gene in tetracycline, BAM-H1 and SAL1? So TBS, can you remember? TBS, for tetracycline resistant gene, we have BAM-H1 and SAL1. And for this, we have APP or PAP, you can remember. Let's remember PAP. So PVU1, PST1, ampicillin resistance, PAP. Can we remember? What are the other molecular scissors present here, or the cloning sites present here? So E. coli 1, CLAV1, HIND 3, PVU2, right? Remember all these restriction sites present here. Is this point clear to all? And what do you understand by ROP? It is the region for, yes guys, what is ROP? ROP is the region for replication of plasmid, right? So this is PBR 322. Now if you want to remove this tetracycline or ampicillin resistance, then you have to insert a foreign gene. So the technique is known as insertional inactivation. Yes or no? Please tell me these points are clear to everyone. This is called insertional inactivation, right? So you should be able to label the things. First thing first, you should be able to identify the locations. This place belongs to ORI. They can give you A, B, C, D and so on, right? This is ORI, this is ROP. In ROP, we have PVU2. Then cloning site in tetracycline resistance gene, it is BAM-H1, and this you will remember. TBS and PAP you will remember. Otherwise, E. coli 1, CLAV1, HIND 3, and ROP has PVU2. Is that clear? And rest you can obviously answer, I know. Next technique coming every year, that is PCR. PCR is for gene amplification. First thing first, the 3 steps. It consists of 3 steps. It occurs in 3 steps. What are the 3 steps in order? The question has already come. Arrange the steps of PCR in the correct sequence. What are the 3? Denaturation, then it is annealing, and then it is elongation. Yes, first of all, learn the sequence. Then what happens in each step? In denature, and guys see, this temperature, denaturation occurs at very high temperature of 90 plus 90 degrees Celsius. Annealing occurs at around 54 degrees Celsius, and extension occurs at 72 degrees Celsius. After learning the sequence, see the temperature fluctuation occurring at every step. Now, denaturation occurs by heating. So you heat it at this temperature and the 2 strands will separate. You're basically breaking the, heating breaks the hydrogen bonds. It breaks the hydrogen bonds and both the strands will separate, and each one will act as a template. Now towards the template strand, now we are going to add these primers. These are the primers. These are the RNA primers. So what happens in annealing? RNA primer is attached. RNA primer is attached. Please tell me these points are clear. Is this point clear? Then after the binding of RNA primer, this is the RNA primer. Now also note down at which end RNA primer will bind. See, replication always proceeds from 5 to 3 dash direction. RNA primer is binding to, primer will bind to, binds at 3 dash end on both sides. 3 dash end of the template strand. That is why elongation will occur in 5 dash to 3 dash direction. Yes or no? So the new strand polymerization will always occur in 5 dash to 3 dash direction. Yes. Now that happens in the 3rd step at 72 degrees Celsius. Now basically what will happen? Complementary deoxyribonucleotides will attach. Yes. Now for the polymerization of these deoxyribonucleotides, what do we need? We need DNA polymerase, and it is a specific DNA polymerase which is Taq polymerase. Yes or no? Now this Taq polymerase comes from, yes, this Taq polymerase comes from a thermophilic bacteria which is Thermus aquaticus. Thermus aquaticus. It lives in, it is found in hot water springs, Thermus aquaticus. So the enzymes of this bacteria are heat stable. Are we clear till here? It can survive temperatures till 100 degrees Celsius or above. So Taq polymerase is what? It is nothing but heat stable DNA polymerase. It is heat stable DNA polymerase. And it is found from this thermophilic bacteria. Please tell me these points are clear. Right? Now, moving on to the next very important process, that is agarose gel electrophoresis. You should remember certain things about agarose gel electrophoresis. Number one, what is the objective? Why are we doing this process? It helps us in separation of the DNA fragments. See, every year we are getting question. Separation, for the separation of DNA fragments. Right? Now you should know that DNA is a negative molecule. DNA is a negative molecule. So it will move towards the positive. Move towards positive. That means it will move towards anode. Anode is positive. Yes. So if this is DNA, if these wells contain DNA, this is DNA, this is DNA. So this side is negative and therefore it will move towards positive. What is the principle? The smaller the DNA moves the fastest. Smaller the DNA, fastest it is, and will reach the anode first. So the one which so it will start separating like this, So the one which will reach the anode first, this one is the smallest DNA fragment. Yes or no? This one is the smallest DNA fragment, and one which is left behind, this one is the largest of all. Yes. So why do we call it agarose gel? Because you are using agarose gel here. Agarose gel is obtained from, this is obtained from a seaweed. Yes or no? Obtained from a seaweed, which is red algae. Seaweed, Gracilaria and Gelidium. Have we read this or not? Right? Now, this gel will have a sieving effect. This gel has a sieving effect. So, DNA is going to move through this because of the sieving effect of the gel. Is that clear to all? Now, how do I study it? Very important. So, in order to visualize the DNA fragments, you have to stain it first with ethidium bromide. Learn the name. Ethidium bromide is the name of the stain or the dye. Right? Now, this appears orange. It appears like bright orange in UV light. What is the color of the bands? Bright orange, not in normal light, but in UV light. Please tell me these points are clear. Bright orange in UV light, asked again and again. Bright orange bands appears. So, DNA appears at bright orange bands in, yes please, in UV light. Is that clear? Fine. All right. So this was agarose gel electrophoresis. And finally, removal of this DNA from the agarose gel is known as elution. From gel is called elution. Are we clear? So you should know the objective, you should know the principles. DNA being negative moves to positive, and the smaller one will move the fastest, and visualization will occur by staining it with ethidium bromide, and it will appear bright orange in the UV light. Are these points clear to all? Next, guys, we have process of rDNA technology. This question has already come up, and you should know the sequence. All the sequences are important, as we know. So first point is isolation of DNA material. So it is isolated from the living organism, and this living organism from which it is isolated is known as source. So for that we use enzymes, right? To remove all the unnecessary things, we are going to use carbohydrates, lipases, proteases, nucleases, RNases, particularly not DNases, right? So we'll use all the enzymes, and finally we will extract DNA in the chilled ethanol. We'll use chilled ethanol to extract DNA. This process is known as spooling. I'm just writing the key words here. Chilled ethanol to extract DNA, process known as spooling. Then we are going to cut DNA at the specific locations, which are known as restriction enzymes. They will cut at the specific location, right? Now after cutting, and we will extract it, we will do electrophoresis here, by the way. Electrophoresis will be done here for separation, and then after separation we are going to insert the DNA into the vector. Now this is a method of transformation. Once it is inserted, it will be known as, so this process will help. These are the methods of transformation. So it can be vector based, or we can do it with multiple other methods also. So here we have used a vector. Otherwise, we can use a gene gun or micro injection, right? And also we have heat shock method. Please tell me these points are clear. So these are different methods of transformation. Anyhow, we have introduced our DNA into the host cell. When it is introduced into the host cell, this process is known as transformation. Now the host has become transformed or transformant. It has become transformant. Is it clear? Now selection of the recombinant host cells by using the selectable markers. Finally, see, after electrophoresis, actually we'll amplify it also. So that is done by PCR. Now see, then selection of recombinant, and then multiplication in the bioreactors, So all these processes, they are upstreaming processes. This is all upstreaming. Finally, when the product will be formed, that product will be separated. That cloned gene is expressed to produce the desired protein. Now this will be separated, and this is known as downstreaming. So you should know what are the 3 steps done in downstreaming process. It is separation, purification, and preservation. Yes or no? Purification and preservation. Please tell me these points are clear to all. You have to remember, I'm just helping you revise these things. These are the points, these are the topics which will come again and again. Next, guys, coming on to restriction enzymes. Restriction enzymes are also known as molecular scissors, right? Which molecule do they cut? They cut DNA. So they are scissors because they cut DNA, right, at specific locations. And these specific locations are known as restriction sites or cloning sites. Yes or no? Why do we call them restriction enzymes? Because they will restrict the virus or any they're found in bacteria. They will restrict the virus from getting replicated, so they restrict it. They are found in bacteria or prokaryotes as a cellular defense, against virus. Yes or no? And against virus. Now let me tell you more about these restriction sites. These restriction sites should be palindromic in nature, right? Let me know in the chat, guys, how many restriction enzymes have been isolated? Yes, how many have been isolated from how many bacterial strains? Fill in the blanks quickly. Those of you who will watch it later, make sure that you answer these questions in the comments. Yes, guys, you have to know the answers. If you are not able to find answers to these questions, 900, very good. From 900, almost 900 restriction enzymes have been isolated from how many strains? Somebody is saying 230, somebody is saying 250. What is the exact answer, everybody? What is the exact answer? What is the number of strains, guys, from which these 900 restriction enzymes have been isolated? One such example, let me know in the chat quickly. So one such example is that of E. coli 1. And the first one, the first to be discovered, was hind 2. Hind 2 was the first one to be discovered from Haemophilus influenza. Now, how do we name them? How do we name them, guys? The first name indicates the genus. The other two letters indicate the species, coli. The third one indicates the strain. And this is the Roman numeral indicates the order of isolation. So it means hind 2 was discovered first, but not isolated first. Yes, please. What is the answer? 230 strains. Absolutely. Answer is 230 strains. Are these points clear to all? Right? Now, whenever we are using these restriction enzyme, the same restriction enzyme will be used for the vector as well as for the host. And they lead to the production of, sticky ends. They produce sticky ends. Generally, guys, now for E. coli 1, there is a specific location. Now each one will have a specific site. For example, E. coli 1 will always act at gut duct. So this is the site. Is that clear? And the restriction site consists of how many base pairs? How many base pairs are present? This restriction site consists of 6 base pairs. Specific for each site. Sorry, the restriction enzyme specific of each site. Yes. Is this clear? Where does it cut? Cuts little away from the center. Cuts little away from center. Are these clear? So between GA, not in the center, because it has to produce the sticky ends. Are we clear? Moving on to the next. That is microbes in human welfare. Every year we are getting questions from this topic. Please go through your PYQs. I challenge if you don't see question from this topic. And even in this year. See, there are 3 bioactive molecules, streptokinase, cyclosporin A, and statins. Streptokinase A is obtained from streptococcus, which is a bacteria. Now this is a bacteria. Streptococcus bacteria. While cyclosporin A, Trichoderma polysporum, and Monascus purpureus, both of them, these are fungus. If any one of you finds it difficult to match and gets confused whether cyclosporin A comes from Trichoderma or Monascus, you learn my name, Miss TC. Can you learn this, Miss TC? Yes? So MS, so Monascus gives us statin and Trichoderma gives us cyclosporin. Think of me and you'll be able to match it very perfectly, right? So Miss TC, so Monascus gives us statins and Trichoderma gives us cyclosporin A. Remember? Okay, so what are the functions? See, streptokinase A is an enzyme with fibrolytic activity, clot buster, dissolves the fibrin clots, treatment of myocardial infarction given to heart attack patients. It's a first aid. Now, cyclosporin A, produced by Trichoderma polysporum, immunosuppressive agent, and it is used in the organ transplant patients to prevent the graft rejection. Statins given by Monascus purpureus, it is a blood cholesterol lowering agent because it is an inhibitor of the enzyme involved in cholesterol formation. See, competitively inhibits that enzyme, lowers blood cholesterol, and hence prevents the treatment of, so given to high blood pressure patients. So it is given to high blood pressure patients. Please tell me these points are clear, right? Now moving on to the next. So these are all the organic acids. Organic acids again very important, and some household products. So first of all, I'll start with these organic acids. See, acetic acid, citric acid, butyric acid, and we have this ethanol, ethyl alcohol. So acetic acid from Acetobacter aceti, citric acid from Aspergillus niger, this is bacteria, this is fungus. Clostridium botulinum, again a bacteria. And then we also have lactic acid, see, from Lactobacillus. But there are other functions. And ethanol, ethyl alcohol from Saccharomyces cerevisiae, it is a yeast. It helps in the production of alcohol as well as in baking. So this yeast is known as brewer's yeast as well as baker's yeast. Now, talking about lactic acid converts milk into curd. Why is curd better? Because of high vitamin B12. So improves the nutritive quality of curd and helps checking the disease causing microbes present in stomach. So curd increases the good bacteria, right? Curd, I've told you already. How is curd produced? By lactobacillus, by coagulation of the milk proteins, partial digestion and coagulation of the milk proteins. For dosa, we need lab and yeast fermentation, and toddy by the fermentation of sap by some yeast. So it is a South Indian traditional drink. Of course, alcoholic. Bread, Saccharomyces cerevisiae. Swiss cheese by Propionibacterium shermani. It is a bacteria. Roquefort cheese, Penicillium roqueforti. It is a fungus. Now, Swiss cheese is characterized by large holes. Why do these large holes produce? Because of carbon dioxide release. Are we clear? Roquefort cheese. Now, these are the organic acids, as I told you, and their sources and their functions. Please make sure that you learn these two tables for sure. Every topic is important, guys, but we are just revising those topics which are 100% appearing in every NEET exam. Is this clear to everyone? Please tell me. Is that clear to everyone? I am moving aside so that you can take the screenshots also. Is that clear? So make sure. Now, let's come on to the diseases. First of all, see, the questions from this chapter, health and diseases, are usually very simple. They are asking you the disease and the causative agent or their symptoms, right? So first of all, talking about the diseases, learn the names of the causative agents. Common cold, rhinovirus. That is why we called common cold as rhinitis, right? Caused by rhinovirus. Nasal congestion, sore throat, cough, headache, tiredness. Affects the nose and respiratory passage but not the lungs. Next viral disease that you should know about, that is AIDS, HIV, fever, weight loss, chronic diarrhea, persistent infections, and severe weakening of immune system, right? Then bacterial diseases, the two which have been discussed by NCERT, typhoid and pneumonia. Typhoid by Salmonella typhi. Pneumonia is by Streptococcus pneumonia and Haemophilus influenza. See their symptoms: sustained high fever, 39 to 40 degrees Celsius, typhoid. And think of typhoid Mary also. She was the classic carrier. Then, other than high fever, weakness, stomach pain, constipation, headache, loss of appetite. In pneumonia, fever, chills, cough, headache, lips, fingernails turn grey to bluish because of oxygen deficiency, and alveoli gets filled with fluid. That is a key point for pneumonia. Talking about protozoan diseases, the two have been discussed. One is malaria and second is amoebic dysentery. So different species of plasmodium are causing the different types of malaria, and the most dangerous one is falciparum. See, recurrent attacks of fever, chills, headache, anemia in severe cases. Amoebiasis or amoebic dysentery by Entamoeba histolytica, constipation, abdominal pain and cramps, stools with excess mucus and blood clots. So excess mucus and blood clots is occurring in amoebiasis. Now see here, in ascariasis by Ascaris lumbricoides, the common roundworm, internal bleeding in intestine, muscular pain, fever, anemia, blockage occurs here. Is that clear? Filariasis or elephantiasis, Wuchereria bancrofti, as well as Malai. Now, filariasis, it causes blockage of the lymphatic vessels, leading to the inflammation and swelling of the lower limbs, such that the lower limb becomes elephant-like. That is why we get the name elephantiasis. Please tell me these points are clear. Is that clear? All right. Now, moving on, let's talk about the drugs. Drugs is again a very important topic. Very important topic. See here. These are drugs. So first category is opioids. Where do you get opioids from? Papaver somniferum, commonly known as poppy. Which part gives you that? It is the unripened capsule. So you obtain latex from unripened capsule. or gives you that latex, and on getting oxidized you get opium. You get opium. So this is the natural one. Then by methylation you get morphine, and by diacetylation you get heroin, right? So by methylation of opium we get morphine, and by diacetylation of morphine we get heroin. So it is chemically diacetyl morphine. Yes, it is a depressant. Remember, it is a depressant. And where are the receptors present? Receptors on GIT and gut and CNS, gastrointestinal tract and central nervous system. That is opioids. Clear to all? Now, coming on to the second type, that is cannabinoids. Cannabinoids are those which are obtained from cannabis sativum, that is the hemp plant, right, which is going to give you a hallucinogen. So in hallucinogen we have two categories, LSD and cannabinoids. See, what are the different types of cannabinoids? So you should be able to identify this leaf. It is given in NCERT. What are the different types of cannabinoids? It is charas, ganja, hash, marijuana, right, bhang. All these names are given in NCERT. And the side effect, they mainly affect your cardiovascular functions. Cardiovascular functions. Is that clear? Then talking about another hallucinogen, which is LSD, lysergic acid diethylamide. This is obtained from a fungus called ergot fungus. What is the class of fungus to which this belongs? The scientific name is Claviceps purpureus. What is the class? It is Ascomycetes, one very potent hallucinogen. Then also remember Atropa and belladonna, Atropa, belladonna, sorry, and Dhatura. They are basically sources of stimulants. They are basically sources of stimulants, but in high dose, it becomes hallucinogens. Yes or no? Let's talk about other stimulants. So one, as I told you, Atropa, belladonna and Dhatura. Then we have tea, we have coffee, right? And then coca. So we have coke or crack or cocaine. So cocaine, it is obtained from a coca plant, that is Erythroxylum coca. It is a bitter white alkaloid, bitter white powder, usually snuffed, that is cocaine. Then tobacco, leaves are used, or Nicotiana tabacum, and active chemical is nicotine, chewed as well as smoked. Please tell me all these points are clear. Is this clear to everyone? Please tell me, guys. Now, components of immunity, another very important topic from this chapter. First of all, see, let's talk about the innate immunity. Innate immunity, and then we'll talk about the specific immunity. Everyone, please tell me these points are clear to all. Yes, please. Are we clear? Give me a thumbs up, all the serious students who are revising with me. Quickly let me know. Quick. Guys, we have already completed 20 topics. We have already completed 20 topics. Focus. All clear? All right. Now, let's revise all the components of immunity. All the components of immunity. First, let's talk about innate immunity here. In innate immunity, guys, we will first talk about the physical barriers. What are the physical barriers? Think before you write. Then I'll tell you the chemical barriers. Then we have cellular barriers as well. And finally, the cytokine barriers. So these are all innate barriers. In physical barriers, guys, we have skin, mucus, hair in the nostrils, and cilia to push out that mucus. Yes. In chemical barriers, we have tears, saliva, then HCl in stomach, lactic acid in vagina, and ear wax in ears. Now both tears and saliva, they have an antibacterial agent called lysozyme. It destroys the cell wall. In cellular barriers, guys, we have WBCs, not all, but the phagocytic ones, which is neutrophils. So phagocytic WBCs. Neutrophils, we have macrophages and monocytes. These are the three phagocytic ones. And other than phagocytic WBCs, another one is TK cells. Sorry, these are NK cells, which destroy the tumorous growths. NK, natural killer cells for tumorous growths. And finally, cytokine barriers. These involve interferons, gamma interferons. These gamma interferons are going to destroy the virus, and they're produced by those cells which are already infected by virus to protect the neighboring cells. Are these points clear? Please tell me. So these are cytokine barriers. Everyone clear? So these are antiviral. Clear to all? Guarantee that questions will come. See, first of all, I have no link with NTA, but the reason I have chosen these topics is because they have always come up. You open your PYQs. If you have solved PYQs, na darling, then you should not be asking these questions to me. I don't know why. At this point of time, you are asking me. Have you never solved PYQs? Have you never seen these questions in the papers? I am scared for you. Honestly, I'm scared for you. Now, this is innate immunity. Innate immunity means which is present since birth. It is non-specific in nature. This is non-specific in nature. Right? And then let's talk about the specific immunity. Specific immunity has few key features. It produces memory. It is specific for each and every organism. Then it can distinguish between self and non-self. Self and non-self, right? And also it has high diversity. You give it as many antigens, it will produce as many antibodies. So we still don't know what is the limit of our immune system. So this specific immunity is known as the acquired immunity or the immune system. What are the two components of this? Major components are lymphocytes, right? There are two types of lymphocytes. These are type of WBCs. We have T lymphocytes and we have B lymphocytes. T lymphocytes are going to provide you cell-mediated immune system. B lymphocytes are going to give you antibody-mediated immune system. Is that clear to all? So these are the components. First of all, know the components. Is this clear to everyone? Is this clear to everyone? All right. Now, the bonus point is you should know about graft rejection, guys. Graft rejection is during the organ transplant. So, all the individuals who undergo organ transplant, have to take immunosuppressants. We've just talked about the immunosuppressant, that is cyclosporin A. Why? Because organ transplant is not really accepted by your immune system, because you're giving a foreign organ to the body, not acceptable. So graft rejection occurs in the case of organ transplant, and that is why you need to take immunosuppressant drugs, right? Now, which are the cells which are responsible for graft rejection? It is the TK or TC cells, lymphocytes. Killer cells or cytotoxic cells, they are involved in graft rejection. Is that clear? And in order to suppress graft rejection, that is why we take cyclosporin A. Is this point clear to all? So see, this time also you got this question from the drugs. You got this question from drugs. This was 2026. You got question on nicotine. You got question on morphine, heroin, right? And then cocaine. These questions were in the match the following. This question has already come up in 24 also, 26, 24. Now, moving on to, these are the components of immunity, graft rejection. Now, let's talk about one more, that is different types of diseases. So, you must know the diseases which come under different categories. We have already discussed the bacterial, viral and all those. Now, let me tell you about allergies. Just know the names: allergic diseases, autoimmune diseases, and when immunodeficiency occurs, immunodeficiency diseases. Right? See, allergic diseases, allergy also known as hypersensitivity, exaggerated immune response. And we know histamine and IgE, that is very responsible. So make sure that you know those names. So histamine, so I'm just writing the keywords, their role. IgE, right? We call it hypersensitivity. So what are the diseases? Hay fever, right? So anaphylactic shock, where the whole body goes in that state, anaphylactic shock. And then we also have bronchitis, right? Asthma, these are all allergies. Hay fever is against sporopollenin. In anaphylactic shock, the whole body goes in a state of shock. Bronchitis, asthma related to your respiratory passage, right? So what is the treatment? You take antihistaminic drugs, you take adrenaline because the body is in a state of shock, right? You take cortisol, that means steroids also. Then coming on to autoimmune diseases. In autoimmune diseases, basically, your body fails to distinguish between self and non-self. What are autoimmune diseases you have discussed? Exophthalmic goiter, yes, in which hypersecretion of thyroxine occurs. Then myasthenia gravis, very, very important. Myasthenia gravis is very, very important. Myasthenia gravis, the acetylcholine receptors on the muscle fiber, they are destroyed. Then we have rheumatoid arthritis, eczema. Yes, please. What other diseases do you know about? How do you get these notes? You can message me on Insta probably. I'll share it there. Clear? All right. So these are some autoimmune diseases that we know about. Also, one more, Hashimoto's disease, in which there is low secretion, hyposecretion of thyroxine. Hashimoto's disease. It is a type of hypothyroidism, right? And then immunodeficiency diseases. In immunodeficiency, immune system is gone, like in case of SCID, which is a genetic disease though. ADA deficiency, right? Severe combined immunodeficiency. And then we have AIDS, while this is acquired, not genetic. Are these points clear? You should know the examples of these diseases under which category. Let's talk about human evolution. You've got this question multiple times. So first of all, we have Dryopithecus, gave rise to Ramapithecus, then became Australopithecus. Then Australopithecus became Homo habilis. From habilis, we got Homo sapiens, Homo erectus. And from erectus, we got the first modern man, that was Homo sapiens neanderthalensis, or simply known as Neanderthal man. Your NCERT does not talk about Cro-Magnon and directly leads to the modern man, that is Homo sapiens sapiens, that is us. First of all, you should know the order. So how do I remember? Doctor, right? So basically, Dr. Ram went to Australia, made it its habitat, right? And then after that we got erectus here, and then Neanderthalensis and Cro-Magnon and modern man. Okay, so Dryopithecus, second part of this topic which you should know, that is the cranial capacity. What is the cranial capacity here? 350 cc. 350 cc become 500 cc. 650 cc. cc is the cranial capacity. Bigger the cranium, bigger the brain, more the intelligence. So look at the cranial capacity. The intelligence is increasing. Here it became 900 cc. Neanderthalensis became, so Neanderthalensis has around 1400 cc. Cro-Magnon had around 1650 cc, and we have around 1450 cc. So you can get the question correct sequence, also match the cranial capacities. If you know the order, you can easily match the cranial capacities also. Is that clear? Now moving on to the examples of homologous organs. Look here. Homologous organs, two examples have been given here in case of plants, the thorns of bougainvillea and tendrils of cucurbita. So what are homologous organs? Homologous organs, they have same origin, so similar structure but different function, but perform different functions. So this is the key to check whether the two organs given are homologous or analogous. Thorns will give you protection, tendrils will provide support, but they are both developing from the axil, hence similar structure, similar origin. If there is, if you have to identify whether they are homologous or analogous, this is the key. Just look out for the function. Different function means homologous. Similarly, see here, the forelimbs of human, cheetah, whale, bat, they have similar structure, same set of bones, but each one of them are using the same set of bones for different function. Human is using forelimbs for multi-purpose, then cheetah for running, whale for swimming, bat for flying. So these are the examples of homologous organs, yes or no? So these are the forelimbs. This is second example given here. Now talking about the analogous organs. In analogous, see, different origin, different structure, but same function. So if the two organs are performing same function, they are analogous, right? Why? Because they are living in the same habitat. Why did they start performing different function? Because they moved to different habitat. So what are the examples given in your book for analogous? Yes, quickly let me know in the chat, everyone. What are the examples of analogous organs? Number one, it is the flippers of penguin and dolphin. Yes. Second, stem tendrils and leaf tendrils. Are you able to recall which plants have stem tendrils, which have leaf tendrils? from morphology. If you are not able to recall, please write it down, find out the answer, or let me know in the chat or in the comments. Which plants have this? I'm asking question from morphology. Now third, potato and sweet potato, both storage of food. Eye of octopus and mammal, both are photoreceptors, but different structure, different origin, but performing same function. Yes guys, are we clear? So these are different analogous organs. One last, wings of any insect, they can write down the name of an insect, and wings of bird. Both are involved in flight, but in insect they are skin modification, in flight they are the modification of forelimb. Are we clear? These are the different examples that you should know. And you should also remember HDAC. What do you have to remember? HDAC. So homologous organs show divergent evolution, while analogous organs show convergent evolution. So what is the evolution? What is the mnemonic? The mnemonic to remember is HDAC. So homologous organs have divergent evolution and analogous organs show convergent evolution. Is that clear? And then these are the examples. How to identify? Just on the basis of function. Different function, homologous. Similar function, analogous. Will you be able to answer these questions? Please let me know in the chat. Next, timeline of various life forms. Now please take a screenshot of this, guys. Please take a screenshot of this. Very, very important. Coming on to Australian marsupials also. Wait. So this is the timeline. You should know this. 13.9 billion years ago, origin of universe. 4.5 billion years ago, origin of Earth. 4 billion years ago, that is 500 years after. 500 years after, that is origin of life. 3 billion years ago, first non-cellular life. 2 billion, first cellular life. 500 invertebrates. 350 jawless fishes. 320 seaweeds and few plants. This is a chronology all given in NCERT in a scattered way. So 200 ichthyosaurus, that is fish-like reptile. Fish-like, yes, ichthyosaurus. Then 65 dinosaurs disappeared. 15 million primates, Drio and Ramapithecus, the ancestors. 3 to 4 man-like primates walked in Eastern Africa. 2 million years ago, Australopithecus. 1.5 million years ago, Java man, that is Homo erectus. And 1 lakh to 40,000 years ago, Neanderthal. There is one more given, agriculture, that is 10,000 years back. Agriculture evolved just 10,000 years back. Are these clear? Quickly take a screenshot here. Now coming on to marsupial radiation. So marsupial radiation or divergent evolution, or we call it adaptive radiation. So common origin moved to different places. See. So we have Tasmanian wolf, tiger cat, banded anteater, marsupial rat, kangaroo, wombat, bandicoot, koala, marsupial mole and sugar glider. You have to remember these 10 names. Which of the following set of marsupials do not show this radiation or adaptive radiation? Which of the following animals do not show? So if you remember these names, only then you will be able to identify. Is that clear? We can easily remember kangaroo. And we can easily remember kangaroo, koala, bandicoot, wombat, right? Then just see, all the other names are double names. Banded anteater, tiger cat, Tasmanian wolf, sugar glider and marsupial mole. Clear? Now, if we have to see the convergent evolution, that is adaptive convergence, also known as adaptive convergence, this is seen in between placental mammals and marsupial Australian marsupials. So first of all, learn them separately also. Placental mammals are mole, anteater, mouse, lemur, flying squirrel, bobcat and wolf, right? And these are separate, and then learn them in pairs also. You have to learn them. So there are 3 types of questions coming from this table. 1, which of the following organisms are placental? Just these names. Which of the organisms are Australian marsupials, which we have just learnt in the previous flowchart also. And then, which of the following set of animals show adaptive convergence? Is that clear? You should be able to answer the question in all the 3 possible ways, right? Remember them in pairs also. Next, talking about transcription, coming on to genetics, guys. See here. So, question has come up again and again this year also. Transcription unit consists of promoter, structural gene, and the terminator. There are 3 components, 3 regions. So, this is promoter, this is the structural gene, and this is the terminator. This whole paragraph was there in 2026, yes or no? Whole paragraph. There were 5 statements. You have to choose the correct ones. Now, see, remember, so which one will decide out of these, which one is going to decide that out of the 2 DNA strands, which one will be the template strand and other one will be the coding strand? So the one which decides is the promoter. Yes, so it is clearly written here, since the 2 strands have opposite sides, and see, it is the right. So see here, first of all, let's read. The 2 strands have opposite polarity. DNA dependent RNA polymerase will polymerize. It will always polymerize in only one direction, that is 5 to 3 direction. Remember. Now, the strand with polarity 3 to 5 is the template strand or the non-coding one, and the other with polarity 5 to 3 is the coding strand. Is the coding strand. Is that clear? So 3 to 5 is the template strand and 5 to 3 is the coding strand. Is that clear? So what is the benefit? So template strand itself is known as the non-coding one. Who determines it? It is the promoter, right? Now 3' end is always known as the downstream and 5' end is known as the upstream. Is that clear? Now what is the benefit of coding strand? See, all the reference points while defining a transcription unit is made with the coding strand. This is the benefit. So if this is the template strand, what is the complementary coding strand? Can you find out? And can you also find out what is the complementary mRNA coded? Now there is a trick. So if you are given the template strand, the mRNA strand will be complementary. It will be complementary. And we know template strand has polarity 3 to 5', while mRNA will have polarity 5 to 3'. So polarity is also opposite polarity as well, right? But if you are directly given the coding strand, how will you transcribe, how will you decide the this mRNA strand? So if we talk about the coding strand and the mRNA strand, both are same. They are same, not complementary. Coding strand and mRNA strand are similar to each other in reading. Just that A is replaced with, T is replaced with U. T replaced with U. And even the same polarity also. We know it has 5' to 3' direction polarity, and same is the case of mRNA. So utilize your time very wisely, guys. Utilize your time very wisely. Is that clear? Clear to all? And now let's read these lines which were here. So promoter and terminator flank the structural gene. That means they're present on the sides of it. Promoter is located towards the 5' end of the structural gene. Was this line there in NCERT in 2026 or not? Questions might be repeated, guys. Questions might be repeated. We never know. The DNA sequence that provides the binding site for RNA polymerase is a promoter, and it defines so promoter defines the template and the coding strand. You switch the position with terminator and the coding and template strand will also be reversed. Next line was terminator located towards 3' end, that is downstream of the coding strand, and defines the end of the process of transcription. Additional regulatory sequences further present upstream and downstream to the promoter. Some properties shall be discussed while the regulation is discussed. This whole point is very, very important. And usually, as compared to other processes, there are more number of questions coming from transcription as per the trend in PYQs. So make sure that you learn this process very well. And then again, I am telling you, if you feel that your transcription topic is very strong, you find translation difficult, or you have to find it out yet, take the papers. How much are you prepared? And to get into that zone, take the tests, guys. Unacademy All India Test Series is still live. You still have there is possibility that you can still take 6 tests before you go for the final NEET exam. So the mistakes you might be making in the NEET exam, you should be making them now while you're taking the test. Get into that zone. Now, talking about the genetic code. See, this is the genetic code here. So what are the properties? It is triplet. Who told us that it is triplet in nature? A physicist, George Gamow. How many codons are there? Total 64 codons are present. Now, out of 64 codons, 6 will code for the amino acids, while 3 are the stop codons. Which one are the stop codons? We know UAA, UAG, UGA. These are the 3 stop codons. Then it is degenerate. Degenerate means some amino acids can be coded by more than 1 codon. Then there is no punctuation, hence it is comma length or continuous manner. Then nearly universal. Nearly universal means it is same for all the organisms. Few exceptions are found in bacteria. So, few exceptions have been found in the mitochondrial codons and some protozoans. That is why we say nearly universal. Then talking about AUG. AUG, guys, AUG has a dual function, codes for methionine as well as it is a start codon. These are the stop codons written here. Is it clear? You don't really have to learn this table. Just some general ones will have already been learned that you know, like CCC, proline, right? You know GAG codes for glutamic acid, GUG codes for valine. So some general, you don't have to waste time in learning the genetic code now. Let's talk about lac operon. Let's talk about lac operon, guys. Here. Now, remember the symbols first. First of all, what are the different type of questions coming from this topic? You should know the symbols. I codes for, I is regulator. Why do we use I as a symbol? I word indicates inhibitor. Now, we know a regulator is going to produce repressor, and this repressor is going to inhibit the process. So that is why we use the symbol I. Otherwise, what does I indicate? I indicates regulator gene. P for promoter, right? O for operator. And these are the 3 structural genes. These are the 3 structural genes. Now, this is in case of a prokaryote, and this is a polycistronic gene. Yes, prokaryotes have polycistronic. So, another very important question. What does each structural gene code for? So, Z codes for beta-galactosidase, Y for permease, A for trans-acetylase. Is that clear? Then, what does regulator codes for? Repressor. Where does repressor bind to? It binds to the operator gene. So, see the sequence. This all happens in the absence of inducer. Who is the inducer here? Lactose or allolactose, sorry. Allolactose. Now, in the presence of lactose, what will happen in the presence of lactose? Lactose is the inducer here. So, lactose will bind to this repressor. So, there was this question once. So, if mutation occurs in this regulator gene, if there is a mutation in the regulator gene, will that repressor bind to the inducer? No. Right? In that case, what will happen? In that case, it will not bind and the process will continue. Is that clear? So, I codes for repressor gene. Now, in the presence of inducer, that is lactose, lactose will bind to it. So, promoter and operator, they are all free, and that is how promoter is binding to RNA polymerase and structural genes are active, Z, Y, A. What do they code? Match the following. Is that clear to all? Now, next is human reproduction. Look at this flow chart, guys. This flow chart has a comparison. There are there's a variety of questions coming from this particular topic. Take a screenshot quickly. Take a screenshot here. Done? So see, first of all, we know be it spermatogenesis or oogenesis, it occurs in 3 steps. One is multiplicative phase, growth phase, and maturation phase. Right? See the sequence, spermatogonia to spermatogonia by multiplication, by undergoing mitosis. This is multiplicative phase. They simply increase their number. Then spermatogonia is growing in size. It increases in size and becomes primary spermatocyte. See the ploidy also. Then primary spermatocyte will undergo meiosis 1 to form 2 secondary spermatocytes and finally 4 spermatids. So this is maturation. When this will undergo meiosis, so what type of division occurs in maturation? It is meiosis. In multiplication, it is mitosis. Now all these processes, they occur at the time of puberty. See, this all starts at puberty and continues, and then continues lifelong. So see, from one primary spermatocyte or even one spermatogonia, we get 4 functional sperms, and there is equal cytokinesis, no arrest. So it is a continuous process. Continuous process starting from puberty, goes on lifetime. Talking about oogenesis, oogonia will form more oogonia, that is multiplicative phase. It occurs during the fetal life. Then primary oogonia will become primary oogonia just by, or we call it, primary oocytes, just by growing in size, right? So when oogonia becomes primary oocyte, there is no change in the ploidy, but it grows in size, that's it. Again occurs in the fetal stage, right? Now, this is growth phase. Then it enters the maturation phase. Now, primary oocyte will start meiosis 1. Okay, it will start it enters the meiosis 1 and gets arrested here in which state? Diploteny stage. So it all occurs in the fetal stage and it is arrested. So maturation will start in the fetal stage but will not complete then. It will be arrested there in the maturation, in the starting of the maturation phase, prophase 1, diploteny stage only. Is that clear? Then it will remain paused for many years before the female enters her puberty. So all this occurs in the fetal stage and then stops. It discontinues for many years and then restarts again at puberty. So it completes meiosis 1 at puberty and forms secondary oocyte and first polar body, which again pauses and waits for the entry of sperm. So secondary oocyte will enter the meiosis 2, will be arrested at metaphase 2 stage. So after fertilization, it will enter into the anaphase 2 and complete meiosis 2. Is that clear? So do we understand this? So ovum and the second polar body. So overall, we have primary oocyte forming 1 ovum. So our NCERT says 2 polar bodies. So that's it. So why is 1 polar body formed? While here there are no polar bodies formed because of unequal cytokinesis. Notice this. So these are all points of differences. Continuously acting, discontinuous. All acting during the puberty and after and continues lifelong. This starts in the fetal stage, pauses, re-begins at puberty and then ceases permanently at menopause. You getting that? So do you see the differences here? So you can get many statement based questions based on these differences. Are we clear point? Yes. Yes, guys. Clear? Now talking about the birth control methods. Yes, please. So we should know. So please revise the birth control methods, all the names and the categories they belong to. See here. Let's have a quick recap. So we have natural methods. We have barrier methods. We have IUDs. We have oral contraceptives, implants and injections. Right? Then we have these emergency contraceptives and we have permanent contraceptives. Are these names clear? So we have natural, barrier, IUDs, oral, implant and injections, emergency contraceptives, and permanent. Yes? Now, what are the three different natural contraceptives? So we have periodic abstinence, also known as calendar method or rhythm method. Yes. Second, we have lactational amenorrhea. First, revise the names, and then we have coitus interruptus. Right? Then in barrier methods, we have mechanical barriers or physical barriers. And we also have chemical barriers. Chemical barriers cannot be used in isolation. It consists of foams and jellies and creams, which are used along with the physical barriers to improve their efficacy, and creams. So they contain spermicidal products, which can destroy the sperm. Clear? While in physical barriers, guys, we have condoms for males, femidomes for females. These are female condoms, and cervical caps, vaults, and diaphragms. Clear? In IUDs, again, we have three categories. We have inert or non-medicated, we have copper releasing, and we have hormone releasing. First of all, you should just know the names of these contraceptives under different categories. So these are non-medicated or inert. We also call them non-medicated. Right? What are the examples here? We have Lippes loop. So here we have copper T, copper 7, and even multi-load. Names before I am speaking. Yes, please. Are you able to recall these names? Then, about oral contraceptives, we have steroidal as well as non-steroidal. In non-steroidal, guys, we have just one name, which is Saheli or Centproman. While for here, we have Mala D, Mala N, right? What is the difference? So they contain either progesterone only or a combination of estrogen plus progesterone, right? So these are steroidal, these don't contain the steroids. Now, as far as implants and injections are concerned, the same combination is found here also, but in the form of implant or in the form of injections. Hence, they have better life, right? So these pills have they are either to be taken on the daily basis or on some special days of the menstrual cycle or once in a week. But implants and injections are taken either once in a month or once in a year or once in 3 to 5 years. So the efficacy is quite longer as compared to these pills. Emergency contraceptives will have the same components, progesterone and estrogen, but in very high level, very high amount of this, right? And also even IUDs within 72 hours can act as emergency contraceptives. In permanent, we have vasectomy for males and tubectomy for females. Is that clear? So which one are the hormonal methods here? The hormonal methods, I'll write down H for it. So hormonal methods are, see, lactational amenorrhea, because here prolactin level will be high in the initial few days of intense lactation, and it will stop the menstrual cycle. There will be no menstrual cycle. So this is hormonal. Another hormonal method here is this. These are the two hormonal IUDs. Then these pills are hormonal. These steroidal pills are hormonal. Implants and injections are hormonal. Emergency pills are hormonal. So these are some hormonal methods. Which is the most ideal method? The ideal method is IUD. This is considered ideal, and even, yeah, this is the ideal method. Most accepted method is either condom or IUD. Again, is that clear to all? Now you should know the mode of action for all, and especially the most important topic is IUD. Most important topic is IUD. So learn the categories, learn the mode of action, but the most important thing is IUD. For example, what is the function of inert IUD? How do these non-medicated lipase loop act? So it basically increases the phagocytic activity, right? It makes the uterus more hostile towards sperms. Then copper releasing. So copper ions, they lower the fertilizing capacity and they inhibit the sperm motility, suppress the sperm motility, right? And then about hormone releasing, how do these LNG20 and progestasert act? They will make the cervical mucus more thick, and they'll make the uterus hostile. Please tell me, and unsuitable for implantation. It will make the uterus unsuitable for implantation. Are we clear with all these points? Please tell me. Yes, guys. Is that clear to all? All the serious students, please give me a thumbs up. Is this chart ready? And are you able to recall all the things? And now coming on to ART, assisted reproductive technology. See here. So we have some in vitro techniques and in vivo techniques also. It will be in vitro as well as in vivo. So in vitro, fertilization outside the body, outside the female body, and in vivo, fertilization will occur within the female body. Right? So basically IVF, ZIFT, GIFT and ICSI, these are all in vitro. These are all in vitro, right? And then we have, so not GIFT, sorry, this is in vitro, this is in vitro, this is in vitro, but GIFT is in vivo, and this IUI is in vivo, and AI is also in vivo. So first thing first, you should know which methods come under the category of in vivo and which are in vitro. Now see, in vivo, IVF or ET, so let's say this is test tube baby program. In test tube baby program, what happens? So eggs and sperm are taken in a test tube, right? And so fertilization occurs in the test tube, and later on they are transferred into the female body. So it depends at which stage are you transferring. Now this embryo transfer can occur at the zygote level or embryonic stage, which is up to 8 blastomere stage. So that is known as zygote intrafallopian transfer. So zygote is transferred into the fallopian tube or up to less than 8 blastomere stage, right? If you go for stage higher than this, then we have IUT, intrauterine transfer. Intrauterine transfer. Is that clear to all? So intrauterine transfer, so stages above 8 cell stage will be placed back into the body, and where? In uterus. So here it is fallopian tube in ZIFT, here it is uterus where it is placed. So what about ICSI? It is intracytoplasmic sperm injection. So sperm is directly injected into the nucleus of ovum. So one sperm, one ovum, right? A single sperm is injected. Now in that case, again we have to follow up it with embryo transfer. So both in vitro techniques, be it test tube or be it ICSI, both of these techniques are followed by ET, embryo transfer, and embryo transfer further has two ways, ZIFT and IUT. Then talking about GIFT, gamete intrafallopian transfer, when is it done? When the female cannot produce egg but can carry the pregnancy. So egg from the donor female is taken and kept into the fallopian tube. Is that clear? Now IUI and AI. AI is artificial insemination and IUI is intrauterine insemination. The difference is here insemination occurs till the vaginal level and here it is little deeper into the uterus. So generally in the low sperm count or in case of erectile dysfunction where the male fails to inseminate the female, in that case these techniques are done. Is that clear? So these are also in vivo. Please tell me these points are clear. This is ART. And now coming on to the sequence of events, guys. Now sequence of events in angiosperms. So you should know what are the steps followed. So starting from pollen mother cell, which is 2N, it will undergo one meiosis to form one microspore tetrad. So there is one tetrad formed, but tetrad will give rise to cells will separate and it will give rise to 4 microspores or pollens. These are all N, right? Why? Because meiosis has already occurred. So all the 4 are functional. So you should know the sequence, right? Now each one of them will undergo 2 mitoses. So one will be vegetative cell, one will be generative cell. So the mitosis occurring here will show asymmetric spindle formation. Right? This is a vegetative cell. Now generative cell. So vegetative cell will help in pollen tube formation. Generative cell will undergo second mitosis. So how many meiosis and how many mitoses in males? 1 meiosis, 2 mitosis. So now this generative cell will give rise to 2 male gametes. Right? So this is microsporogenesis, this is microgametogenesis. Clear? Alright. Now look here. In megaspore mother cell, again 1 meiosis giving rise to 4 megaspores. Now out of these 4 megaspores, only 1 will be functional, 3 will degenerate. 3 degenerate. Now the one which remains functional, this one functional megaspore, it will undergo 3 mitoses. Now these mitoses will not involve cytokinesis, hence we call them free nuclear endosperm. Sorry, free nuclear divisions. Free nuclear divisions, and it will give rise to this 8 nucleated embryo sac. Now later on this embryo sac will undergo maturation to form a mature embryo sac. Now what does this mature embryo sac look like? We'll talk about this. So how many meiosis? 1. How many mitoses? 3. 1 meiosis, 2 mitoses, 1 meiosis, 3 mitoses. Is that clear? See here. So this is the structure of ovule. So a question is repeatedly asked around hilum. So what is hilum? Hilum is a junction between. What is the junction between? It is a junction between the funicle and ovule. Right? Funicle is the stalk. Micropyle is the opening. Micropyle, by the way, is known as the upper end. Chalaza is the lower end. Is that clear to you all? Now, there are 2 integuments, nucellus. What is the ploidy of nucellus? N. What is the ploidy of each cell in embryo sac? N. Nucellus is 2N, by the way. Nutritive tissue. So, embryo sac. Questions are asked again and again about embryo sac. Embryo sac is the female gametophyte because it contains the female gamete. Now, see here. This is the micropylar end, first of all. This is the chalazal end. 3 cells. How many antipodals? 3 antipodals on the chalazal end. What is the ploidy of each? N. Now, 1 egg cell and 2 synergids again on the micropylar end, and together they form the egg apparatus. So you should know the composition of egg apparatus. What is the polarity? It is towards the micropylar end. What are the components of egg apparatus? One egg cell or the female gamete and two synergids. Then synergids always have filiform apparatus. What is the function? To guide the pollen tube. They release some chemicals to guide the pollen tube. So pollen tube is going to enter through one of the synergids, right? So they guide. And what about the two cells? So these are also haploid, N plus N. Don't write it 2N because they are separate. Two polar nuclei in the central cell. So what is so special about the embryo sac, the female gametophyte? It is female gametophyte because it contains the female gamete. So embryo sac, mature embryo sac, guys, is 7-celled and 8 nucleated. So we have seen 8 nuclei, and why are the 7 cells? There are 3 antipodals, one egg cell, 2 synergids, and 1 central cell. Yes, so 7 cells, but nuclei 2 because there are 2 nuclei here, so it becomes 8. Are we clear, everyone? Now, next, guys, coming on to human physiology. So take a screenshot of this chart, guys. This table will help you revise the functions, the source of all the hormones. Yes, please. Quickly take a screenshot, everyone. Take a screenshot. So it has hormones from all the different glands and the functions as given in NCERT. The date of the exam, Indu. So you will do that right before going for the exam. Is that clear? All right. Next, see this table. Take a screenshot, everyone. Take a screenshot here. So revise this table. It will help you revise all the functions. Now coming on to the parts of the brain, guys. So we have 3 parts: forebrain, midbrain, and hindbrain. Forebrain has cerebrum, diencephalon, and it also has, by the way, olfactory lobes. It also has the olfactory lobes responsible for smell. Now midbrain, and then we have hindbrain. See, forebrain, the largest part is cerebrum, which is large, convoluted, largest part divided into 2 cerebral hemispheres. And what are the functions? Seat of intelligence, memory, thinking, imagination, interprets the sensations and coordinates the voluntary actions, and responsible for learning. Clear? Now talking about the second part, that is diencephalon, lies below the cerebrum, and it consists of thalamus, hypothalamus. You can skip epithalamus here, by the way, but that will form your pineal gland later on. Now see, thalamus, it is a sensory relay center. It is a relay center. Hypothalamus, it is a link between the nervous system and endocrine system and controls the pituitary gland, maintains homeostasis, regulates body temperature, hunger, thirst, fatigue, biological rhythms, and even some emotions, right, like sexual desire. Then coming on to olfactory lobes, skip this, write down olfactory lobes are important for sense of smell. So the organisms with well-developed olfactory lobes, how many? 2 olfactory lobes. The well-developed olfactory lobes will give you better sense of smell. Now talking about midbrain. So midbrain is situated between forebrain and hindbrain. It has visual and auditory reflexes and controls the movements of head and this head, eyes, eyelids, pupil, and so on. Then we have hindbrain with 3 parts: cerebellum, pons virili, and medulla oblongata. This is the second largest part of the brain. Very similar structure, like 2 cerebral hemispheres, we have 2 cerebellar hemispheres. The function is voluntary muscular activities, posture, and balance of the body. Pons virili, again a relay center and helps in respiration. It has respiratory centers. It has respiratory centers. Talking about medulla oblongata, the lowermost and continues with the spinal cord. See, involuntary activities, heartbeat, breathing, blood pressure, and reflexes of swallowing, vomit, coughing, sneezing, all these are in medulla oblongata. Now remember, in brain, guys, in brain, there is white matter and gray matter. So gray matter is on the outer side and white matter is on the inner side, but not in case with medulla. Medulla and spinal cord, they will have different arrangement. They will have outer white matter and inner gray matter. Are these points clear? So make sure that you learn these functions from this table and revise them. Is that clear to all? Yes, guys? All right. Now, moving on to last 10 topics left, guys. Now, let's talk about synapse. So, synapse is a junction. Synapse means junction between the two neurons, right? It could be a junction between muscle fiber and the neuron, or it can be a junction between two neurons. Now, we have basically two types of junctions. One is chemical junction or chemical synapse, and we have electrical synapse. See, first of all, a synapse consists of three components, which are a presynaptic membrane, synaptic cleft, and the postsynaptic membrane. Now, in case of chemical synapse, there is a chemical involved that is called a neurotransmitter. That is called neurotransmitter, right? So that is why we call it chemical synapse. So see, in chemical synapse, the synaptic cleft is filled with these neurotransmitters, and the receptors are present on the postsynaptic membrane. This is also a question where the receptors present. Is that clear? So we have presynaptic membrane, we have synaptic cleft. Cleft means gap, which is fluid filled though, and then we have postsynaptic membrane. So synaptic vesicles, they store the neurotransmitters. They contain neurotransmitters. So they will be released from here into the synaptic cleft and bind to the receptors on the postsynaptic membrane, and that is how message from one neuron will be passed on to the next neuron. Of course, there is so much of process involved in release of transmitter, binding of it to the next neuron. So it is a more time consuming process. But if we talk about electrical synapse, this is a quick way of transmission. It is quick. This is rather slower, right? Why quick? Because here between the two, see, this is pre and this is post. So in between the two, there is a gap junction. So the message flows along this wall to the next. So it behaves like one common neuron. Is that clear? So these are the two types of synapses, and you should be clear with the differences between them. Now, another, you have to learn this chart. You should know how many bones are present in your cranium, guys. Your cranium has 8 bones, right? Your facial bones are 14, and in total, they form your skull. So skull has two components, cranium and facial bones. Hyoid is not a part of skull, remember, not a part of skull. How many? 1 bone. Then you have auditory ossicles, that is ear ossicles, 6 bones, malleus incus. Then vertebral column has 26 bones, sternum is 1, ribs are 24, further divided into true, false and floating. So we have first 7 pairs, that is 14, which are true, then 6 which are false, 8, 9, 10, and then 11, 12, that is 4 ribs which are floating in nature. So you should know the proper distribution of each. Then coming on to appendicular skeleton. So this has 80, appendicular skeleton will have 126 bones, pectoral girdle will have 4 bones, 2 clavicles and 2 scapula, and then each limb will have 30. So total 16 upper and 60 in the lower. Then unlike pectoral, which has 4 bones, pelvic will have 2 bones. These are coxal bones. These are 2 coxal bones. Each coxal bone is formed by the fusion of ilium, ischium and pubis, right? Is that clear? Each by the formation of, so we have 2 ilium, 2 ischium, 2 pubis. Clear? Now see the bones in the lower limb and bones in the upper limbs. So be very clear with the names. Second very important aspect of this chapter is the joints, guys. Very important. So first of all, we have non-movable, immovable, that is fibrous joints, then partially movable, cartilaginous, and completely movable, we have synovial joints. So no movement here, right? So examples are sutures. So sutures, they're present in the cranium. What are they formed of? We also call them fibrous joints because they're made up of dense fibrous connective tissue. Then talking about the connective, so there is partial movement, there is limited movement here, right? Where is it present? In between the vertebral column and also between the two coxal bones, pubic symphysis. Now, very important and the most important are the synovial joints. Freely movable because of the fluid present here, that is synovial fluid, right? So allows considerable movement. So first is ball and socket, then we have hinge, then pivot joint, then gliding joint and saddle joint. You should know the examples of each. They can come up for the exam in match the following also. Is that clear? Will you be able to revise all of them? Yes. Is that clear to all? All right, guys. Now, next is the process of muscle contraction. We have process of muscle contraction. This question has already come. Arrange the following steps in the correct sequence. So there was a sequence based question here. So see, first of all, a neural signal arises. It releases neurotransmitter. Which neurotransmitter is involved? Acetylcholine, right? So, and where does this neural signal arrive? Neuromuscular junction. Which nerve will bring that message? A motor neuron will bring that message. Is that clear? Then action potential reaches the sarcolemma, the muscle fiber, then what will happen? Calcium ions are released into the sarcoplasm. So where is calcium ion present? Calcium ions are present in the sarcoplasmic reticulum. They release the calcium in the sarcoplasm. Now this calcium binds to the troponin and it removes the masking of active sites for myosin. Is that clear? Now what will happen? Utilizing this energy, ATP hydrolysis will occur on the myosin head and it binds to the exposed active sites on the actin to form a cross bridge. So see here. So these are all steps. So what is happening here? So when ATP hydrolysis occur, so then cross bridge formation takes place. Why? Then when when ADP leaves the head, then the head moves backward, leading to sliding. And finally, when ATP is regenerated here regeneration is occurring, this leads to the breaking of the cross bridge. Is that clear? Now, during that process, guys, this is muscle contraction. So, know these steps. Is that clear to all? So these are the steps. Now, what are the changes that occur in the sarcomere? Sarcomere is the unit of contraction. See, so what happens to the sarcomere? So Z line pulls inward, comes closer, right? Then shortening of this leads to shortening of the muscle, that is contraction. Now, in this, I band reduces, A band remains the same length. Remember that. So what all happen? Z line shortens, Z line come closer, I band reduces, H zone will also reduce. The only thing that will not change, that is your A band. A band remains the same. These are the changes in the sarcomere. Is that clear? Very important disorders. Out of all the disorders, these disorders have a special importance in the hearts of NTA. So see, myasthenia gravis, we have muscular dystrophy, arthritis, osteoporosis and gout. Myasthenia gravis is autoimmune, right? Muscular dystrophy is genetic. Is that clear? What happens in myasthenia gravis? So basically, there is destruction. It is an autoimmune disorder where ACH receptors are destroyed. ACH receptors are destroyed. Just told you. ACH receptors destroyed, affecting the neuromuscular junction. So where are receptors present for ACH? In the neuromuscular junction. So that is why it is affected, leading to fatigue, weakening and paralysis of the skeletal muscle. Clear? Then muscular dystrophy, progressive degeneration of skeletal muscle will take place. Now it occurs because of the absence of a protein called dystrophin, important for contraction. Tetani. rapid spasms, irregular contractions actually due to low calcium. It can be because of low calcium or it can be hormonal also. Then arthritis, arthro word is for joints, inflammation of joint. Osteoporosis is age related, decreased bone mass, higher chances of fracture due to decreased level of estrogen in females after menopause. And gout, accumulation of uric acid, again inflammation of joints. Clear to all? Now coming on to this chapter of excretion, RAS mechanism. Again, NTS love, right? So see, RAS mechanism stands for renin, angiotensin, aldosterone system. This is RAS. So whenever, guys, see, whenever the blood pressure decreases or the glomerular filtration rate decreases, this stimulates the kidney. It stimulates the kidney to produce renin. Is it clear? So renin is produced by kidney. Which particular cells? Juxtaglomerular cells of kidney. Now from the kidney via blood, it will be put into the blood. Via blood, it enters here, and angiotensinogen, which was actually produced by liver, but in the inactive form, renin is going to convert angiotensin into angiotensin 1. Now there is again another enzyme is angiotensin converting enzyme coming from lungs, which converts angiotensin 1 into angiotensin 2. All this happens when BP falls, right? So overall, this whole technique is this whole process is to uplift the blood pressure, which has fallen below the normal level. Is that clear? So angiotensin 1 will change into angiotensin 2. Now see, angiotensin 2 is a powerful vasoconstrictor. So when vessels will constrict, area will reduce, pressure will automatically increase. That is the first function of angiotensin 2. Second, angiotensin 2 is going to stimulate the adrenal cortex gland. Adrenal cortex gland to produce aldosterone. And we know aldosterone is a mineralocorticoid. It will increase the water and sodium retention. And again, when water and sodium retention are increased, blood pressure will automatically increase. Is that clear? One more, angiotensin can also take help from ADH. So ADH will further help in increased absorption of water, so which will further increase the blood pressure. Are we clear? So this is RAS mechanism. So this happens when blood pressure falls, GFR falls. But what if blood pressure is high? For high blood pressure, we have ANF, natriuretic factor. This checks the RAS mechanism. So atrial natriuretic factor, it will check RAS. Check RAS means it will stop RAS mechanism. So nobody is going to vasoconstrict. Rather, ANF is a vasodilator. Just opposite to that. RAS and ANF, they are totally antagonistic to each other. Is that clear? It checks RAS mechanism and therefore helps in reducing the blood pressure. So this is stimulated when BP is very high. It stimulates RAS, ANF. They are totally antagonistic to each other. Is that clear? Now types of nephrons we should know. One is cortical, second is juxtamedullary, just 15 to 20%. Rest 80 to 85% are these. So what are cortical granules? See, cortical granules, this is your kidney, the cortex area, and this is medulla, right? So you should know what all portions. So we have Bowman's capsule, PCT, right? Very small loop of Henle here, and then DCT in cortex area, right? While there are other nephrons where PCT, a long loop of Henle, and DCT is present here, right? Now do you see the difference? So these cortical nephrons, they have small loop of Henle, no vasa recta, no loop of Henle, no vasa recta, so no water reabsorption, no water, not really reabsorption, no water conservation through loop of Henle. So no counter current mechanism here, counter current mechanism. Clear to all? While in juxtamedullary, guys, in juxtamedullary, it is just the opposite. We have long loop of Henle. We have long and well-developed vasa recta. So it is involved in counter current mechanism. Please tell me these points are clear to all. Yes, please. Are we clear? Are we clear here? Yes. So these are the types of nephrons, and you will see a PYQ, one very important figure, very important figure. Direct figure has come up, or you can get questions on the basis of what all things are getting. See, these are your blood vessels. These are your blood vessels, right? Capillaries. So from blood, see, from blood into the nephron, what is this? This is tubular secretion. From nephron into the blood, this is reabsorption. This is PCT, this is DCT, this is loop of Henle, this is collecting duct. Now see, what components are getting reabsorbed from PCT? Bicarbonate, NaCl, nutrients, water, potassium. What is secreted here? This H positive and ammonium ions. Then in DCT, sodium, water, bicarbonate, almost the same. And then potassium and H positive are being secreted here. What about loop of Henle? Descending loop of Henle is permeable to water, and impermeable to electrolytes. Is this line clear? And these lines have directly come in your previous year questions, right? So water will come out. Therefore, descending limb of loop of Henle, this is concentrating limb. Yes or no? This is concentrating in nature. While the ascending limb, guys, you see, ascending limb is permeable to electrolyte. Sodium will come out of it. It is impermeable to water, just the opposite nature. Impermeable to water. Yes. So if water is not coming, sodium is coming out. It means ascending limb is the diluting limb. Yes or no? Right? So what is being absorbed by ascending limb? It is urea. So urea and sodium chloride, these two molecules, these two chemicals are actually important for developing the concentration or osmolarity in the medulla. So as we go down into the medulla, deeper into the medulla, the osmolarity increases from 300 to 1200. Why? Because of these two compounds. Yes. So now see, maximum reabsorption here, least in the loop of Henle, least reabsorption in loop of Henle, and in DCT it is only conditional or facultative. That means if needed, not otherwise. Are we clear? Please make sure that you learn this table very carefully. And then talking about valves in the heart. Yes, guys, valves in the heart. So we have two types of valves, AV valves and semilunar valves. AV valves are tricuspid and bicuspid. It is present between the right auricle and the right ventricle. This is between the left auricle and the left ventricle, also known as bicuspid valve. Now, the closure of these valves cause the first heart sound, that is love, right? Semilunar valves in the beginning of aorta and pulmonary artery. The valves in the beginning of aorta and pulmonary artery, they are semilunar in nature, 3 flaps, right? So this is at aorta, at the beginning of aorta, and that is in left ventricle, while this is at the beginning of pulmonary artery in the right ventricle. So their closure leads to the production of second sound, that is dove. So this is first heart sound and this is the second sound. Remember, both the sounds are produced during ventricular systole. Both sounds produced during ventricular systole. This is during the beginning, in the beginning, while this is in the towards the end of ventricular systole. Please tell me these points are clear. Yes, guys. Hello. Are we clear, guys? Till here? So these are valves. Questions are asked around valves in heart as well. And one very important topic that is ECG. ECG stands for electrocardiogram. Right? This is the graph. This graphical representation is called the graph. This is called the gram. This graphical representation is called the gram. The machine, the device used is called the graph, and the technique is called the graphy. Now see, there are few waves here. Now what are these going to indicate? P wave is going to indicate the atrial depolarization. It is not the same as atrial systole. This graph represents the electrical activity, not the mechanical activity. Right? After P wave, the atria will start contracting. Now, QRS complex indicates the ventricular depolarization, and ventricular systole will start occurring after the Q wave, and then T wave is for ventricular repolarization. Now, the number of QRS complexes is equal to the number of heart beats, or it is equal to the heart rate. Is that clear? Please tell me these points are clear. Now, you got a question from this topic, a very tricky question. What was it? So they asked you not the direct percentage, they asked you to calculate the number of WBCs based on this percentage. So learn the percentage here. Monocytes 6%, right? Eosinophils 2%, yes. Then lymphocytes 20 to 25%. What about neutrophils, the most abundant? They are yes please. Neutrophils most abundant, they are 60 to 65%, and then basophils are 0.5 to 1%. So they'll give you a specific number and ask you to calculate. Can you calculate? Yes. So all these 5 percentages should be clear, and just to learn them in the decreasing order, the answer is null mab. Neutrophils, lymphocytes, monocytes, and acidophils and basophils. Acidophils is also known as eosinophils. Is that clear? Be sure about the picture and the order and the percentage. Very, very important, oxygen dissociation curve. See, in oxygen dissociation curve, we will see there is a sigmoid shaped curve, right? So in any sigmoid shaped curve, we have this lag phase and log phase, and then the stationary phase, right? Now this curve will show the right hand shift also. This is the right hand shift. It can also show the left hand shift. So left hand shift and the right hand shift. What does this right hand shift indicate? It favors oxygen hemoglobin dissociation. And where does it occur? That is at the tissue level. Then what about left hand shift? Left hand shift favors oxygen hemoglobin association. So oxygen and hemoglobin will associate where? At the lungs level or alveoli level, right? So what are the features which favor the association? Now see, in the alveoli, what are the conditions which favor? High PO2, low CO2, lesser H positive, and lower temperature. These are going to favor, these are all going to favor the left hand shift. Is it clear? Now, similarly, there are factors which are going to favor the dissociation. So that occurs in the tissues. See, low PO2, high partial pressure of CO2, high H positive concentration and high temperature. So they favor the dissociation. Is this point clear? So you have to remember these points, these features, which factors will favor, which factors will be against the association or favor the dissociation, and where do these conditions occur. So overall, the oxygen dissociation curve is going to study the effect of all these factors. Is that clear? So very, very important factors. And now moving on to respiratory volumes and capacity. We have tidal volume, guys. Tidal volume is during normal respiration. Both inspiration and expiration both are normal. So value is 500 ml, and in 1 minute it will be 6000 to 8000 ml or liter per minute. So see the value. Now talking about inspiratory reserve volume. In inspiratory reserve volume, inspiration is forceful while exhalation will be normal. What is the value here? What is the value here? Value is 25 to 3000 ml. Then talking about expiratory reserve volume. In this, expiration is forceful, but inhalation will be normal. So value is 1000 to 1100. So match the following, decreasing order of arrangement, so many things. Residual volume. It remains in lungs even after forcible expiration. Value 1100 to 1200, slightly more than ERV. Then coming on to inspiratory capacity. This is TV plus IRV. Expiratory capacity, it is TV plus ERV. Functional residual volume. So that is ERV plus TV. And vital capacity has three components: ERV, TV and IRV. While total lung capacity has these components: ERV, TV, RV and also IRV. So you can get the question in this from this topic with respect to the numericals, definitions, values. All these questions can come up from this topic. Is that clear to all? Right? And next topic is that of heterophily. Quickly see this question, guys. So the topic of heterophily, very, very important. Just a second. Yes, please. Very, very important topic of heterophily. So heterophily is an example of plasticity. So basically, this is the ability of the organism to show different kinds of structures in response to the environment or the phases. Is that clear? This is plasticity. One example of plasticity is heterophily. It is heterophily. Now, three names: heterophily in cotton, coriander and larkspur. CLC. In cotton, coriander and larkspur, in these three, it shows as per the different phases. So juvenile phase has this and adult phase has this. This is an example of larkspur shown here. While with respect to different shape and shape of the leaves with habitat difference, so look at the shape of the leaf in the terrestrial habitat and in the water habitat. Example is buttercup. Only one example given for heterophily in terrestrial and water habitat due to environment. This is heterophilous development due to environment. Are these points clear? So very important examples. And then coming on to ETC. First thing first, you learn the names of these complexes. Complex 1 is NADH dehydrogenase. 2 is succinate dehydrogenase. 3 is cytochrome BC1. 4 is cytochrome C oxidase. And the fifth one is ATP synthase. This is complex 5. So direct question with match the following can also come up. Is that clear? Second question that you should be ready with: how many H positive ions can be pumped out through complex 1? 4. From complex 3, 4. From 2, none. From 4, 2. Is that clear? How many components of this and what hypothesis is being followed here? Chemiosmotic pathway. Is that clear? Now, hardly a paper where you don't get question from the C3 cycle. First of all, the sequence of the process: carboxylation, reduction, and then regeneration. So, sequence CRR, reduction and then regeneration. Then, see, one CO2, and this is a 5 carbon compound. So, you should get 6 carbon here, but no, you get a 3 carbon compound, but they are produced in 2 molecules. So 2 molecules of each are produced here. Again 2 molecules, and then 1 carbon will be removed here and 5 carbons will be regenerated here. Is that clear? Now during carboxylation we don't use any ATP or NADPH, while during reduction 1 ATP 1 NADH is utilized for the reduction of 3-PGA2 triphosphate 1. But since we have 2, so we'll multiply this also as 2. Clear? And then again during regeneration 1 ATP is used. So for the fixation of, so if we talk about fixation of, for fixation of, see, 1 CO2 molecule, 2 ATP and 2 NADH used during reduction, and 1 ATP during regeneration. So in total 3 ATP and 2 NADPH, right? But 1 CO2 is not going to produce glucose. So for the production of glucose, you need to fix 6 molecules. 6 such cycles have to undergo. So that means how many? 18 ATP and 12 NADPH are used. Is that clear? Now this is a C4 cycle. So what is the difference? See here. In C3 cycle, guys, this step, that is carboxylation, this uses Rubisco. So carboxylation is catalyzed by Rubisco, right? Now here and there, in the C3 cycle, this is C3 cycle, right? In C3 cycle, this Rubisco is present in the mesophyll cell, right? While here in case of C4 plant, so these are in C4 plants. In C4 plants, first the C4 cycle will occur, followed by C3 cycle. C3 cycle is mandatory because food will be produced during C3 cycle. Now see, this is the C4 cycle occurring in, mesophyll cell, and C3 cycle occurring here, fixation by Calvin cycle. Is that clear? So mesophyll cells will have another enzyme which is known by the name peptase. So PEP, phosphoenol pyruvate, is the primary carbon dioxide acceptor, while in case of C3 plant, who was accepting, this is RUBP, primary CO2 acceptor, right? The enzyme is Rubisco, while here the enzyme is peptase, and this PEP is a primary CO2 acceptor. PEP has 3 carbons. This is 1. Total it is going to form OAA, which is a 4 carbon compound. That is why the name of the cycle is C4 cycle. Transported, it gets converted into a malic acid or aspartic acid, transported, undergoes decarboxylation, and again forms C3. Now this CO2 will help in undergoing the C3 cycle. Now Rubisco, which will be used here in the C3 cycle, is not present in the mesophyll cell. Instead, it is present in the bundle sheath cell here. Is that clear? Now take a screenshot, guys. This is cell cycle, different stages and the processes occurring. So you have to remember these stages. You have to remember these stages. Direct questions will come up. As far as this prophase is concerned, look here. The prophase 1 consists of 5 different substages. Chromosomes condense, leptotene. They undergo synapses, zygotene. They undergo crossing over, pectine. Then this it undergoes desynapses in dikinases. And so just a second, here dissolution of synaptonemal complex, this is dissolution which will lead to desynapses, that is in dikinases. And finally we have, sorry, this is in diplotin, and finally complete desynapses will occur when it will terminalize. So terminalization of chiasmata, that will occur in the dikinases. So remember the keywords. Okay, so chromosome condensation, leptotin, synapses due to synaptonemal complex in zygotine, crossing over between non-sister chromatids of homologous chromosome specitine. Then after crossing over, it will be chiasmata formation and dissolution of synaptonemal complex, that will be in diplotin, and terminalization of the chiasmata, that will be in dikinases. So see the ploidy of the daughter cells in each phase and learn them. Take a screenshot, guys, take a screenshot, and we will move on now. So with this, guys, we'll complete today's session. Rest of the things, there are some one-liners that I want to discuss with you that I'll take on Saturday. On Saturday, we will revise just one-liners and we are ready to go for the exam. So make sure that still, if you have any problem, please leave your comments, let me know. And also, before we go, guys, you still have opportunity to find out your shortcomings and work on them. But that can only happen if you are ready to take these overall tests. So make sure that you subscribe, you enroll for this test series and understand what your shortcomings are and work on it. Thank you so much. All the very best. Don't lose your heart under any situation. Fight it and win. All the very best.